So the first speaker in our next session is Ethan Orr, and he is the Associate Director of Agriculture, Natural Resources, and Economic Development and Cooperative Extension at the University of Arizona. And he's going to talk about water irrigation efficiency, or the Water Irrigation Efficiency Grant Program.
I really appreciate the opportunity to be with you, and I appreciate that we're going right through the break and just getting into the program. This program is something that's brand new from the state legislature, and it's a combination of both application and research with the goal of becoming one of many tools to help not only save water, be more efficient with the recognition of the biological constraints that we have in both plant and soil health, but also really at a macro level to get more water behind the dam. That's ultimately what we're trying to do. Basically, the way that I look at this is that there's two main components. There's benefits for the growers, and this is the change of a mechanical system, and there's research. Now, I used to be, and still am, an economic developer, and so I would never recommend to a grower, and no grower that's actually in business would say good incentive is a reason to do a bad business decision. So we want to support good business decisions, and we want to do everything we can to kind of incentivize that risk. It was said earlier, Mark Smith said it quite aptly, we are all in this together. We're just trying to work together to, again, conserve water, put it behind the dam, and this tool will apply to some people, this tool will not apply to everyone. As I go through these rules, there is a committee that is made up of research experts. I will say, we know that these rules are not for everyone. These rules were embedded in the legislation. We have tried to change that. We have made recommendations to do that. There is a component in there that's asking for a system that can do a 20% efficiency gain. I recognize in the valley that I see some of the smiles. We already do laser leveling. There are some crops that this will not apply to. One thing that I do have to say, this is a political reality we all face. Most of the legislators come from an urban environment. We have to work with those legislatures to get the money through this, and they started at a 40% efficiency, and we talked them down to 20%. So these are rules that are in place. They will be very effective in places like the Mesa. They may not be as effective in the Valley. As we go forward in the next legislative session, that is a conversation that I think you should have with our elected members. So I want to put that caveat on that. This tool does not apply for everyone, but the research component does. So kind of the history, we have a lot to be proud of. Arizona has a great deal to be proud of. We use less water now than we did in 1962. We have a crop production that is higher, and we have a population that is 3 times what it was in 1962. So this program is really a continuation of kind of the historical legacy, building on the historical legacy of this state. Now, one thing that's very important to me as I start talking about this, this program is applicable for people that own fields, growers, or people that are leasing the fields, but I am trying to leverage this as much as possible. So, for example, we have our first application coming again, 50 acres out of the Yuma Mesa. I want to replace the mechanical system. I want to study the biological impacts in terms of soil stress and plant stress, but then I also want to leverage that with our conservation districts, the NRCS and other funds, to see if we can do some very basic things like better head gates and flood drainage ditch lining to create even more water savings. The $1 million that we have to incentivize research, and this is something Paul and I have had a number of good conversations about, $1 million doesn't go very far. We all know that. But $1 million of seed money to support partnership with industry to incentivize a larger federal grant is quite effective and quite important. So what we're looking for are strategic investments with both industry and university researchers on our experiment stations and other places around the state, other partners, So what we're really looking to do is partner with you to leverage so that this $1 million isn't just $1 million in research. This $1 million draws more money into our region. The same with the application. It's not just about replacing a mechanical system and generating water savings. It's about studying how we can do that. So for example, some of us are at 85% efficiency. We need to study, can we get to 90% efficiency, or is that just not realistic? So we're going to try and put as much water behind the dam and put as much water retained in our aquifers as possible, but we're also going to be studying ways to do an even better job of doing that. Basically, again, the 20% was set by legislation. We tried to change it. We can reimburse the person in the field, whether you're leasing or growing, any crop, if you can show that 20% efficiency, we can reimburse you for the mechanical system up to $1,500 an acre. And so that is enough for a gravity-fed grip. That's almost enough for a center pivot. There are a lot of things you can do with that. We just need to show the thing. What this will probably end up doing is going from a flood to a drip, and we recognize that we have to do more with the salinity. Basically, one of the main things that we're trying to do is partner with you to not only do the application, and we're taking applications now, but to do the research component. So I see a lot of researchers here. We want to work with you to look at the mechanical, or the biological aspects of the soil health and the plant stress. We're going to be doing all of that. And then the last thing I want to talk about on this is another program that we have created, and these are all incentives for the growers. We also have launched from last year an ag apprenticeship program. This speaks to what Tanya was talking about, where we now have 30 people who are part of a different program. The legislature gave us $1 million to fund workforce development. So we're really trying to address both the water issue and the soil health issue and then the workforce issue. And what we've done with this program is we recognize that workers, if you look at this, in the 1900s, 90% of us were involved in the production of ag. Now, certainly less than 2% of us. We recognize that we're not going to be able to replace worker with worker. We're going to have to replace a worker with someone through applied technology who can do the job of 3 or 4 people. And so in addition to this, we're looking for partnerships with growers that you can host an apprentice that we can reimburse you for, that can really go into a job, learn applied technology, and then become a good worker for you. And so these are really the 3 programs that I just wanted to share with our industry partners. The ability to reimburse, we have $23 million that we are ready to work with you to partner, to create water savings, to really avert the catastrophe that we've all sort of alluded of what happens if we don't put enough water behind the dam and we go into dead pool. That is an application, and that is something that we want to partner with you on right now to study that system. We have $1 million that we want to partner with our own faculty and our industry partners to leverage to draw down other dollars from USDA or NIF and other things to create incentives and to create studies on what happens with salinity, what happens with plant stress, what happens when we cut that water, and what is that biological limit to these mechanical systems. And then we also have an incentive program to help you hire, train, and retain good workers. And so we can reimburse people, industry right now, for hiring an agriculture apprentice, helping train them, and turn them into a good employee. So those are 3 programs I just wanted to touch upon. I really appreciate your time. We are here to serve you at Extension. Thank you.
Yes, sir. Does it have to be mechanical?
That's a good question. The system itself has to be mechanical. However, some of the efficiency gain can be through changing crops. So, for example, if you were to move alfalfa, and this is something I'm actually talking in another region, if I were to move alfalfa, move it to a center pivot and change it with a malt barley, you're saving about 2 acre-feet. That efficiency gain that we're documenting is going to be a combination of mechanical and changing crops. But there needs to be, we can pay for the mechanical system, there needs to be a change in mechanical system.
What for just eliminating flushing, which is 30%, which was stated?
If you can show me a 30%, if in your application you can show me a 30% efficiency gain doing that, we can talk. But I'm not saying we would do it, but that's why I have people that are smarter than me on the committee. But what I need to show is a 20% efficiency gain that's embedded in the system. So that's really what we're looking at. I know there was a conversation at lunch of what happens if we just take an on-field, a channel ditch that I have and I line it. I know that Tanya and I were talking about 7 years ago, the return channel was lined. And so there is data on that I'm sure we can get from Bureau of Reclamation and I'm sure we can apply. This is candidly a brand new program that we're standing up. We have a lot of political realities, hence the 20%, but within the political realities that have been created, I want to work with you and we all want to make a way to make this work, if that makes sense. You're welcome.
I can tell you, Debankur and I, the next speaker and I, are so happy that soil health has become so sexy and such a topic, of today. That it's getting the attention I believe, that it really deserves. So I mainly talk about this now. I do get out in the fields, but these are the people, my lab manager. So I'll just call them by name: Sam, Kirsten, Shelby, Chance, Brooke, EJ, and Jamie are really the ones responsible for this work. And I'm also putting up some of the funding agencies and partners that you'll hear about as I go through here that have been really instrumental. And I was able to put together some different work on compost, but also soil health assessment and management, which you'll hear about from Debankur as well afterwards. So how do I look at the world? I'm a soil ecologist by training. I think about plant-soil interactions. I think about the abiotic environment and how it affects soil organisms and plants. I also think about food webs, which oftentimes doesn't get enough debate. Like, what are the organisms that are eating fusarium and fungi, for example? So I think this is soil ecology really was a way for me to enter into agricultural research. So what I'm going to talk about and just give some brief case studies on are really the goals are to make better compost, better assessment of soil health, and better management of soil health. So I'm going to talk about compost first. So I've been working with the main composting company in Tucson just because it's close. I know talking with Robert and others, I'd like to work more with composters closer to Yuma. They have one of the best mascots ever, or whatever, this tree guy called Tank's Green Stuff. And I've worked with them using, for example, biochar, which I'll talk about later here in a second, which is a form of charcoal you make under oxygen limiting conditions, and it's a very stable form of organic matter. It acts as a sponge for water, but also for nutrients. So we worked with this company. They actually added in biochar during the co-composting process. One interesting note, it sped up the composting process, so they're actually able to increase efficiency by making the compost blacker. It heats up more. And my student Shelby looked at the effects of this, of biochar and co-composted biochar. So this untreated biochar means that you add the raw biochar and compost in the field. Co-composted means you combine them and kind of charge up the biochar before you add it into the field. And we found, and we had, as far as I know, this is one of the only studies where there's been a biochar treatment and irrigation. And we went from business as usual, the wettest condition, to pretty damn dry, basically, and did not find positive effects of biochar. This is wheat yield, and except under really the mid and high. So low, you can consider maybe a 50% reduction in irrigation. The mid was in between. So I'm just showing you one piece of data, but this is an example of how we're working to maybe improve the biochar to act as a fertilizer. Under the driest condition, there was no effect of biochar. Our hope was that when you dry it out, and because biochar acts as a sponge, you should be able to add less water and maintain productivity. But that was not the case. So biochar, and I'm finding a lot, is snake oil in some of our soils. We've also looked at using rocks and minerals to slow down decomposition. A lot of what we try to do in my lab is figure out how do you slow down decomposition from compost. Because in our environment, it's hot and wet, right, in an irrigated field. So our compost burns off way too quickly. So we've tried adding minerals, which also, like biochar, help absorb nutrients, and have found, for example, we also looked at basalt from up near Flagstaff and some green sand, some large-scale mineral sources, and did find when you add in these minerals, it does tend to, for example, slow down phosphorus release, which I view actually as a good thing. It's acting as a more slow-release fertilizer. So in the future with compost, I'm looking through some NRCS funding to look at more of a survey approach of chemical and biological properties of compost, as well as application rates. For example, if you add more compost, do you necessarily get more carbon? So far, the answer I see is no. It just still decomposes. So there must be something about, and I'm also very interested, as I've talked with many of you, about crop nutrient density, not only yield, but density, nutrient availability, and through collaborations, I'm getting into pathogen suppression. So we need, soil health means different things to different people, so it's really knowing, understanding for a given farmer or for given conditions what soil health means. I would argue that salt does not affect soil health. Salt is soil health. That is an aspect of soil health. Okay, in terms of assessment of soil health, I've been a part of some synthesis papers. I helped lead a special issue in soil biology and biochemistry on biological indicators of soil health. And I'm very interested in these, if you go to the Soil Health Institute, for example, they give you this long list of mainly physical and chemical properties. Like if you went to the doctor, here's what you need to measure, right? And this is what health is. But the problem is a lot of these are irrelevant, or there's also missing indicators in desert soils. There's also, you can see, really a lack of biological indicators. I think that's where there's the biggest need for understanding soil health. So as an example, we found so far that looking across many different vineyards in the state, Matt, we need to talk later, and about effects of does the enzymes, if you go to some of these soil health assessments, they say measure the enzymes. Well, we found that they are useless. The enzymes do not help understand nutrient availability. You should just measure the nutrients themselves. We've also been looking at more long-lasting forms of organic matter, the ones that last rather than months or so. These last potentially decades or centuries. Mineral-associated organic matter that I have down here. I'm very interested in these sticky substances produced by EPS. Called EPS, that are substances produced by microbes. And then we're also finding that the organic to inorganic carbon ratio is an important predictor of how much organic carbon you can store in a system. And if you're interested, I can tell you more about that later. So going forward, I really want to add to this other category that only has crop yield, really, so far. And I think what we need to add to that, especially here in Arizona, are things like not only the yield, crop nutrient density, the water use, fertilizer use, food safety, dust pollution. So when you fallow a field, for example, does that increase dust pollution? So we need to account for these the pros and cons. So to finish things off in my final 3 minutes or so, I want to mention better management of soil health. So I had a postdoc work on a project in Pinal County looking at the difference between micro-irrigation and flood irrigation. So this is a different place. I liked Hank's use of the word personality, right? The personality is different than in Yuma, where here irrigation was effective. It actually increased crop growth using less water. But what we found here also is that we looked at organic carbon, and just to orient you, these are 3 different depths, 0 to 5 centimeters, 5 to 10 centimeters, and these are the deepest soils on the bottom. And then the rows are a native ecosystem, so this is a reference ecosystem, very common for Arizona, 0.25% organic carbon, right? California on a bad day has 2% or something like that. So we have very little carbon to begin with. With drip irrigation, there was actually a very large increase, about a 4-fold increase in, this is after, I don't know the exact number of years of drip irrigation, but it's been multiple years. We found higher organic carbon with drip irrigation than with flood irrigation. I don't know why yet, but this is clearly showing that if we can show multiple benefits of these different irrigation types. And organic carbon, hands down, maybe besides fusarium, is the next biggest thing we hear from farmers. They want to understand, you want to understand how do I increase organic carbon? Because that's kind of, if a lot of everything else falls into place if you can build up organic carbon. We also found that the drip irrigation, to maybe get more mechanistic, was, I'll just get to the main point, is that the drip, which are these yellow ones, was actually favoring for gram-negative bacteria and fungi, which can maybe handle the drier conditions on average. It might be if there's salt accumulation, maybe there's something about salt accumulation. So I want to understand also the biology. I've talked with Betsy and others and other biologists that I really want to play with others to better understand what's going on there. So the big issue in the room with water, the dream, if you go to a soil health conference, they say, thou shalt maximize soil cover and always have continuous living roots, right? But that requires water here. That's maybe more of a Midwest concept that if you just let it be, you can just grow something without watering it. But here, the reality, of course, we know is that, just as was mentioned by Ethan, like we're having to pay farmers to use less water. So we have basically no non-growing season here, except when you're getting the fields ready for vegetables. And cover and secondary crops are grown during the hottest and driest times of the year here, versus in other places they're usually grown in the coldest time of the year, where water isn't as much of an issue. So we have major problems here, and I've already been working with JV Farms and got one of those seed grants mentioned before from YCEDA to compare bare fallow, sorghum, Sudan grass, and wheat and its effects on soil health. I also want to quickly mention that I'm ramping up for a large-scale comparison, comparing fallowed to continuous cropping, and just to do a more holistic assessment of its effects on soil health. So that's going to be, right now the assumption is if you fallow, as we've talked about, you decrease soil health. But there's other aspects of soil health besides salinity that we also need to measure. And from this preliminary study, we found an interesting effect that the Sudan grass tended to increase mycorrhizal fungi, at least as measured using this PLFA measurement. And it was interesting because that also had lower amounts of really, I would call, leaky phosphorus. And I think it's because the mycorrhizae are holding on. We can view that, what if we can use microbes to hold on to nutrients during the off season and then re-release them later on? So using microbes as nutrient storage as well. So I'll end there and just will say, this is really where I'm going here, better compost, better soil health assessment, and better management. I'd be happy to answer any questions.
I was at a talk a couple of weeks ago with somebody who works in a very, very cold part of the United States. Was looking at the genes that are turned on when plants are in frozen environments and looking at plant survival in frozen environments. And so I asked her, are these the same genes that you can see in drought? Because the bottom line is, in both cases, water's being locked up, or water's not available. And it turns out they are. So what can we learn from all of the work that's going on in most of the United States that deals with freezing conditions? And can that short circuit information for us to find genetic pathways for drought tolerance or drought resistance or drought dealing with?
It seems like, that could be a really great avenue that we don't tend to think about. I guess I more look at it the other way, where we're learning things. We're like the canary in the coal mine for other places as they get drier. But you're right. We can definitely learn from other stressful areas. And I think that, so one of the companies I put at the beginning, is a microbial inoculant. There's more and more of these. Some of them are definitely snake oil, but microbes that you add to the soil that are often developed in other parts of the country. And but I wonder, just how well adapted they would actually, survive around here. But I think these genes, which again is really outside of my wheelhouse, but I think the cultivars, and so forth are really important as well. Yes.
Is there any need of measuring total elemental composition in soils or any of your material, like fertilizer? I'm talking about, let's say, total iron, total phosphorus.
Great point, because most of what we do with nutrients is some sort of an extraction, right? And it's, in my lab, we tend to use the Malic-3 extraction, which is like a plant available nutrients. I don't know. I don't think total actually matters, especially things like phosphorus. A lot of it can be bound up by calcium. So I don't think that total is as important. I think the most carbon and nitrogen are probably the main elements that matter in terms of total pools.
Next we have Debankur Sanyal, and he is an assistant specialist in cooperative extension. He's an associate professor in the Department of Environmental Science, and he is going to give an intro to the Healthy Desert Soils Initiative. Sorry, I did read that before I said it. Debankur is a relatively new faculty member, and we've seen him a bunch of times in UMass, and we're really happy to have him.
Hello, everyone. Thank you for inviting me here, and thank you, everyone, for staying here this long. It's been a long day, and thank you, Joey, for making my job easier, right? Because he already explained most of the stuff I'm going to talk about. So, great, thank you. Well, we all know why we care about soil health. We all have different ideas, but they're already converging into a same direction. Making our environment better and still being able to produce better crops, more crops, making more profit. Because if it is not financially sustainable, it's not sustainable. So both financially and environmentally sustainable system and soil health might be the key. Well, the principles are same, like Joy mentioned, like USDA, NRCS, any soil health institute, any organization kind of recognize that certain principles we have to follow, and it can be followed everywhere in the world, but the techniques, tools, methodologies should be different because it's specific to the site, specific to the problem, specific to the goal of the farm, right? So let's go forward. And if we follow these principles, these 5 principles like soil armor, maintain living roots, diversity, we might get some co-benefits like conserving moisture and having a chance to minimize chemical use, which is not very good for our health, right? Because we'll be eating the food. Well, we all know what our problems are, right? We don't have enough water, we don't have enough carbon, and we have a lot of salt, and it's pretty typical for any desert soil. So we have to figure out a way, because most soil health research that has been done yet, kind of not ignored, but there are less work done on these aspects when you have problems with salt, too much salt, or very less carbon. So literally most of soil health parameters that are recognized by all these institutes, they are dependent on carbon. If you have very, very less carbon, how can you be sure that you are measuring proper soil health? So we have been working on it. So the approaches can be anything, but the principle, the goal, the target will be the same, like minimizing soil disturbance. We have to figure out a way how we can minimize the disturbance, how we can give the ecosystem, the soil ecosystem, a better chance to improve itself. It's a natural process. We see weeds everywhere, people complain about it, but probably that's nature's way to introduce biodiversity. So you're probably lacking biodiversity. Things like that. We have to think, and that's why I'm here to talk with you, to discuss with you, right? Amendments, soil amendments. Well, this can be applied to the soil that might have a potential to improve carbon, biology, but you also need to have a proper environment for the biology to flourish, right? And that comes with better residue management, having a soil armor. I'm not talking about cover crops. Armor can be any type, can be residues, can be cover crops, can be a secondary crop. It's giving the soil a chance to build and not exposing it to erosion processes, right? And then diversity. We have many crops, many options, and that's a great thing to have in Arizona. Previously, I was working with systems with having 2 or 3 crops at most. But here, so many options, and it's kind of forced, but people choose where the market is, where we can make more profit, and how much water we have, with how much water a farm has, right? And this is a cool photo, probably not for Yuma right now, but we started doing this kind of stuff, trying to figure out how we can control weeds in our organic system. can we graze the weeds to kind of repeatedly kill the potential of the weed seed bank? Things like that. Generally, in most of our studies, we try to figure out, okay, what our soils have, what our environment needs, and then figure out what we should do. But it doesn't work really because we have to think about the people who are doing it, the farm. We have to figure out what the farm wants, what they have available, and what they can do with that resources, because we can come up with idea like, oh, let's go no-till, buy a no-till planter, and get a roller crimper, will be fine. But not every farm has the opportunity to do that. So you have to think about that first, what the farm wants, and then kind of design our plan to improve soil health, right? So I started with a basic approach, needs assessment. I did that, and thanks to Robert and Blaze and all these people who helped me out to spread the news, and thank everyone who took this poll. I think we had around 110 candidates, and so we knew, some things we knew, that we will find that water is the most important thing, and we are in desert, right? Happy to see that we are at the end on research, extension, education. We are doing probably a good job. So we are at the lower level, but we still want to make it 0 and have more education programs going on. Salts and soil pH, definitely an important factor. There are certain things, it's natural in this environment. We cannot control climate, but we can work towards modifying it and modifying its impact, right? So in my program, I'm trying to do that. And well, it's good to have, we are hearing this term soil health since the morning. Thank you so much. And it feels so good that people are talking about soil health, and it's not new here. So as you can see in the rating list, so we started with a survey, statewide survey. We went to 43 different farm fields and collected soil samples, worked with Dr. Joey Blankinship with his lab, and Arizona Cotton Growers Association actually funded this project. We looked at all these things. Good thing is, I don't know, but it looks good. I don't know if I was biased only to the good, healthy soils or not. I mean, see the mean soil organic matter, it's almost closing 3%. I don't know. I don't know. Maybe we have very good soils that we sampled. We're going to do more work on that. Another thing is, well, soil organic matter is not probably everything. So there's a term called POXY, which is active carbon. Not many people like it. I don't like it either. But the numbers are saying something. There are some fields having 0 POXY, which is an active carbon part, which is microbially available part of the carbon. So we might have a lot of carbon that microbes are not able to use it. So we are looking into that now. And I think Joey mentioned about the inorganic carbon and organic carbon ratio. So all these new parameters that we need to think about. And this is a project funded by YCEDA. Thank you, YCEDA, again. We are working with, and you might have seen me emailing you a couple of times that I want to get more samples. So we are doing a good soil versus bad soil, and we're asking the growers to give us an idea on where you have a good soil, where you have a bad soil. Side by side, we're taking samples, measuring. It's on process, and this is my technician and Robert collecting. I'm just the photographer. I don't do any work. They do all the work, and I'm very happy to have them in my team, and Robert is a great help. So we are seeing some good numbers. Let's see here. The POXY levels are still low. I would like to look at it around like 350 somewhere. That's a good one. But we're still searching. There are other parameters we'll be measuring. Soil protein, one parameter that we'll be measuring very soon. We have the ability now in my lab. With that, I want to stop here because, like time, it stops here. We need time to build soil health. It's not a one-year game, and I respect that it's a very tough industry. It's very tricky to wait longer because you might miss the opportunity. But soil is a system in itself. It's very complex. No one understands it fully yet, I believe, and it takes a little bit of time, but we are here to work with you to figure out to look at the indicators. So we see the changes happening, but it might not be in the form of getting you better produce, better production yet. It's going towards it. So it's a long-term process. You just have to work together and figure out what is the best way to improve your soil health in your own farm. And I'm here. Anytime you can text me, call me, email me. I'd love to hear. And I have a new project that we are starting right now. We're looking at effect of sanitizers on soil health. And I just started working with Dr. Rock's lab. So I'm very excited about it. We are probably sampling tomorrow again. So thank you everyone for giving me the time. I will take any questions, or maybe we'll move forward. I'll take any questions if you have.
Is the presence of heavy metals is there any concern in the soil health, the presence of heavy metals?
Yes, definitely. Anything as soil, we call it a soil health, but if you think properly, it's also water health, also air health. So heavy metals can be nutrition health. Heavy metals might move to the fruit or the produce that we are going to eat, right? So it's definitely a concern. Yeah.
Did you measure heavy metal?
No, not yet. But if we hear some problems like that, I would love to measure. Yeah. But it's not traditionally a soil health indicator. It did not get that badge yet. But we'll measure if there is a problem. Yeah.
And the heavy metal composition is the total, right?
Yes. Or we can start with extractable, but then total. Yeah. Because extractable is something we are interested in because that probably plant is going to uptake. Total, it might be there, but it's not impacting the plant's uptake, right? So, okay, thank you everyone.
Our next speaker is Pauli. He's an assistant professor in the School of Plant Sciences, and his talk is titled Genetic Improvement of Lettuce Through Phenomics.
Thank you for the opportunity, Stephanie and Paul, to present the research that we've been doing in Maricopa, but on lettuce nonetheless. Just a precursor, my lab mainly is focused on heat and drought stress tolerance in crop plants, including cotton, sorghum, and lettuce. So what I'd like to talk to you today is the impact of data on agriculture. So most of you are familiar with precision agriculture, which is using sensors throughout the field, through the system, the tractors, everything to collect more data to basically improve crop yield and your efficiencies. So typically, better information, most humans, if they have better information, make better decisions. Not always true. But we also know that more efficient practices leads to increased profit margins. And finally, with respect to crop breeding, which is what I'm focused on, better information leads to better selections and improved varieties. So I just want to give a little bit of background here about phenomics, and specifically how data has impacted plant improvement. So phenomics is really the study of traits that define the organism, form, and function. Why you care is because you eat it in this case. This is what we care about. It's the final head of lettuce. It's what you're selling. It's what people are consuming. So a lot of phenomics is associated with new technologies and platforms to collect these data, and specifically large amounts of it. And why that is because genotype data. So genotyping came about in the 90s. Sequencers are cheap to build now. You can generate a ton of sequencing data pretty fast. If you look at environmental data, we're pretty good at understanding how our environment functions, how to measure parameters like temperature, precipitation. And so finally, the last piece was really understanding what that phenotype is, so how the genetics interact with the environment to give rise to what it is we see in the field. And basically, over the last 10 years, we realized we don't have the same scale and depth of phenotype information to really leverage our genomic information that much, nor our environmental information. And why this is critical is we think about the future. We know our environment's changing. We know there's a water crisis. So we really understand how that phenotype changes in response to the environment. Therefore, we can identify the genetics that will give us improved varieties. So I have the unique gift of being able to run the UA field scanner. It's this giant robot that's claimed to be the world's largest agricultural robot. It covers about 2 acres of cultivated land. The data volumes are unprecedented. On an average day, we're generating about 1.5 terabytes of data. Per day, we can do 10 terabytes if we turn on everything. This robot can run around the clock 365 days a year. And so what that means is we are developing some of the highest resolution data sets on how plants grow and respond to their environment. And so in the white box that's emitting the red light here, we have a suite of sensors that include an RGB camera, so just a regular color camera. We have a thermal camera, which gives us the actual temperature of the plants it's imaging. We have a fluorescence camera. This measures how efficient the plant is at converting sunlight into biomass or what you consume. We have a 3D scanner that gives us a three-dimensional representation of the plant, and finally hyperspectral imaging, which measures the wavelengths from 350 nanometers all the way up to 250, basically outside the range of human visibility. And these different sensors give us a holistic picture of how the plant is growing and responding to the environment. The QR code is if you'd want more information on this. The Wall Street Journal did a piece on it that kind of explains more about some of the capabilities of the system. So what I'd like to talk about specifically today is how we're leveraging phenomics research for lettuce improvement. This is our trial field in 2019. In this image, we are growing 300 different genotypes of lettuce. This is replicated about 12 times. So our current project is a USDA SDR grant with Richard Mitchell-Moore at UC Davis, Robert Masson, and there's about every lettuce research group in the U.S. is on this project. It's only $4 million, but really the focus is on understanding the genetics that control key agronomic and stress-adaptive responses in crop plants. So basically looking at this graph, thinking that the blue line is basically well-watered conditions, the red is drought. How does the plant's growth change in response to that environment? And with our scanner, it's not just across the entire season, it's a daily. So how do small fluctuations in growing degree days impact growth? Which varieties respond to it better, worse? And so with that, we'll really get this high fidelity image of how plants are responding to the stress itself. And so the focus of the project here is identifying the genes and the alleles that confer this tolerance. And the secondary part of the project is taking some of these basic science research tools and translating them into production agriculture. Overall, we're working to safeguard lettuce production against climatic variability. I'll be up front, we're playing the long game. This isn't anything that's going to happen in the next few years, but probably 5 years. So some of the populations, the germplasm that we're using. We use a diversity panel of lettuce that includes over 200 different varieties from every market class. Why we do this is because this assemblage of lines represents all the genetic diversity for cultivated lettuce. This means if there's a gene in there that impacts performance under drought, heat, we have a high chance of finding it. The other thing that we're using is recombinant inbred line populations. These are populations that have been developed specifically to answer questions. Our first year we used one on water use efficiency. I forget what the cross was, but basically it's segregated for carbon isotope discrimination, which is how plants keep their stomates open. What we just planted yesterday was a cross between Seriola and I think it was iceberg that segregates for root phenotypes. So this year we're taking it below ground to understand how rooting architecture also impacts plant performance. All these lines have been sequenced to about 6x coverage, if not more. So that means as we find these phenotypes, we can associate them with the genetic markers that can be used by breeding companies to target specific segments of the genome to move those into already used commercial varieties. So what's interesting about our system is because it's such a precision thing and because we collect so much data, we can track each individual plant in the field across the season. We can map up every single sensor data with individual plants. So in the slide, you can see here's the whole image, just a small section of the field. You can see the actual lettuce heads being identified. We use a ton of machine learning and artificial intelligence to do all this automatically because, again, there's no way you can sit there on these spreadsheets and do this because we are talking about terabytes of data. So these same lines are identified over time. And so each individual head of lettuce is tracked, and we use this because this allows us to develop these time series of understanding plant growth. So this data set's unique in that probably nobody else in the plant science community, from corn, soybeans, neither bigger row crops, has data like these. And basically we can understand and track each head of lettuce knowing its microenvironment. So this field has been soil characterized on a 3D depth. So we know what the environment is. We know how much water the plants are getting. We know how much nitrogen, sunlight. And what this is allowing us to do is get a very high resolution, again, of understanding what genes and alleles are impacting plant growth in response to the environment. So that's kind of, that kind of information allows us to take it to the next step where we can study the individual plant. So we have our traditional phenotypes that we use, like height, length, width, volume, that give us a very, gross definition of the morphometrics of the plant. But some other things that we're working on is topological analyses. These are finer structures within the plant itself. So this can represent how leaves, basically envelop around the head of lettuce if it's iceberg. Romaine, like the compactness of the head. This allows us to target traits that are typically not scored in field trials. And so with this information, again, it's allowing us to use the phenotype data to improve our breeding, to basically better target these varieties for what the market's after. So some of these data, it's like, well, how do we actually use this for production situations? When we first started, we had this, drone imagery. We had an algorithm that basically went through the images, could tell you roughly what size the head of lettuce was. We showed it to a grower. He said, that's cute. Don't waste my time with it. I was like, fair enough, sir. And he says, Duke, there's people out there harvesting lettuce by hand. If it's not a big enough head, they're just not going to cut it. So who cares about a map? He's like, what I really want you to do is I want you to predict what that yield's going to be a month in advance. He's like, that's something I can use. So I said, oh, okay. Challenge accepted. And so we've developed these algorithms that take in environmental information, data about the plant, how it's growing and responding. And what we can do is basically, as we start to see, collect these earlier data points, we can use the model we've developed to predict what the final yield will be, in this case, 4.5 weeks out from harvest. In this case, this is one of our better cases of 99% accuracy, not bad. So this is something we're still working on developing. Again, it's nice to be here and understand if this tool has any relevance. Maybe it doesn't. I hope it does. We can also combine this type of plant predictions with our genetic information for doing enhanced breeding through genomic selection. We also look at canopy temperature to understand which plants are more efficient with their water. Basically, which plants can maintain economic yield with less water. One of the final things that YCEDA supported when I first started, and thank you, Stephanie, was helping us acquire a thermal drone. And Fusarium wilt is just another type of osmotic stress, right? The pathogen blocks up the xylem, the plant's not moving water, it gets warm. Turns out it shows up really nice in aerial imagery. So, we're flying these production fields that YCEDA provided to us. We have guys on the ground, they're going out, they're measuring individual heads of lettuce with the thermometer. We can fly the drone. We can find the heads that are infected with, Fusarium. Why this is exciting for me personally is it's also an application tool for management. We have a group from ASU that does drone swarm technology. They're like, yeah, we can deploy this on a swarm of drones to map out large areas, identify, map, and report that back to growers. From a breeding standpoint, it allows us a fast way to screen trials and basically identify lines that are harboring some level of resistance to the pathogen. And finally, I just kind of want to end, I can play that. So with this gantry, we are generating a ton of data. When I first started, I met Mark Smith, and he said, Duke, this is great. You got to move to Yuma. And I said, no, I live in Tucson. And he says, fine. I'm going to take your gantry, I'm going to uproot it and move it to Yuma. So Mark, this is my solution. So we've taken our entire field, and we've built the first virtual reality field environment. So in this imagery, it's not that high res because it takes too much computational power to render the plant color as well as all the plants in the field. So this is just a small subset, so it'll play. But so you put on the goggles, you're cruising around the field, Mark. You can go up to any individual head of lettuce, select it. You can pull up all of its data for the entire growing season. So its growth curve, its thermal response, the individual point clouds, and you, as the expert, can tell me if it's interesting or not. This is the only way that we can get through 700 terabytes of data. There's no way. The spreadsheets are literally a terabyte. Excel won't even open them. And so this is one of the best tools that we can think of right now for working with our data. So we're working with NSF on a new program that they have for data-human interactions. And so I would say, again, to the industry experts, I don't know lettuce that well. I'm just trying to fake it. But if I can get this tool, put this in your hands, and you walk through our field and tell us, that's cool, that sucks, that's super powerful for us. And that helps us kind of tailor what we can do for the industry. Oh, I think there's one more slide. So I guess it's just the conclusion. So, the scanner itself is a very unique tool being applied to lettuce. Realistically, it's probably one of the cutting edge projects that we have. People are super excited about it. But again, identifying key genes and allele for breeding, and developing improved varieties that deal with less water, deal with higher temperatures. We are looking to help improve agronomic practices that impact production. And finally, I would just say that I'm here to help.
YCEDA that people have been talking about, that's our seed funding program, and you can get more information about that on our website. It's desertaggsolutions.org. Our next speaker is Kerry Cooper. He's an assistant professor in animal and comparative biomedical sciences. And his talk is investigating the evolution and survival of foodborne pathogens in different desert agricultural environments.
All right. Well, thank you, Stephanie. Good afternoon, everybody. I don't have cool 3D pictures. In fact, I get to talk about everybody's favorite subject, which is whole genome sequencing and foodborne pathogens, which I know gets everybody excited. So I'm going to give everybody a free puppy at the end of the talk. Just kidding. So I'm going to talk about one project that was funded by the CPS that led into another project about all of this. So it may take me a few minutes to explain how we had to set all this up in order to do this, but I'll walk you through all of it. So the goal with this project overall was to determine the mutation rates that the foodborne pathogens were accruing in potential different agriculture environments. In particular, we were targeting soil and irrigation water. And so the idea that this was kind of built on was that I'm sure anybody that's in food safety knows that the FDA has Genome Tracker, which was built as a way of tracking potential outbreaks, but also anytime they get samples that are positive, they sequence it, put it into this giant database with the idea that foodborne pathogens, for the most part, tend to be regional. So if you get a hit somewhere and you check the database and it matches something in a particular region, then you can start to hopefully narrow in on that, start asking more in-depth questions in the epidemiological investigation, and hopefully get everything product off the shelves and the outbreak ended quicker. So the question becomes, though, if a pathogen is identified and cultured and put in the database, but it remains in that environment for 20, 25 years, moving around, circling through different environments, it's going to accrue mutations. Well, that may potentially then lead to false positive hits or may change that so that the genome tracker doesn't work. So that was the principle behind this, was to look at, okay, if they're out there for a long time period, how many mutations do they accrue? If they're cycling through different environments, like they're moving from soil into additional soil, or they're moving from soil to water, or soil into animals and things like that, and then into water, how are they going to accrue mutational, how many mutations are they going to accrue? So we looked at 3 different pathogens, kind of the 3 major bacterial pathogens. E. coli 0157, which was the isolate that we used, was from the 2006 spinach outbreak. Salmonella typhimurium, which was from irrigation water in Salinas, California. And a Listeria monocytogenes strain that was from a 2014 stone fruit outbreak. So they all had some type of fresh produce association with them. We looked at, as I mentioned, different environments, soils from leafy greens fields, irrigation water that were water supplies for those leafy green fields, and we also had a control to kind of see what it does when it's not in some type of agricultural environment and to kind of assess the mutation rate there. In this case, it was buffered peptone water. So obviously, we couldn't do this in fields, so we did this in my laboratory. We set up these microcosms. So the soil and water came from actual agricultural fields around here in Yuma, but they were not. They were brought back to my lab. They were tested, confirmed they were negative for pathogens, and then we set up these microcosms in plant growth chambers, which the plant growth chambers allowed us to then control the temperature, the humidity, as well as the amount of light that each of these microcosms was getting. So we set it to the average temperature, the average humidity, and light for each month of the growing season. So we started it in September based on the information from our industry collaborators for the Yuma. The one thing that's missing here is that we also were doing this for Salinas, but I'm not going to talk about that one, obviously. And then we inoculated them. We inoculated them with an extremely high level, 10 to the 8th per gram or mil, which is never, hopefully we're never going to see that out in the agricultural environment. But the idea was we wanted a large pool in order to detect these mutations. So we had, as I mentioned, long-term colonization. So that meant we put the pathogens in, and then we just left them there, and we would sample them every couple of weeks, but they would just stay in those environments all the way through. The original goal was to go for an entire year or 52 weeks, but then a little pandemic happened, and that kind of threw us off a little bit. So we wound up going for 42 weeks within there. The other thing that we did was cycling. As I mentioned, we did cycling within the environment. So cycling meant that we did it for 2 weeks in soil or up to a month. After the first couple of months, we changed it from every couple of weeks to a month, just for logistics. So we cycled it soil, cultured it, put it back into soil, cultured it, put it into soil, and cycled it that way. Same with irrigation water. And then we also did soil and then into irrigation water, then back into soil, in trying to mimic it moving around in between different environments to see. And so, as I mentioned, we cultured it the first couple of weeks for the first 3 months, and then once a month for the final, all the way through week 42. So what did we find? We found a couple of things. One, the good news was that early on, from September to December, the pathogens wouldn't even survive for 2 weeks under Yuma conditions in either the soil or the irrigation water, although they were perfectly happy in the control, in the buffered peptone, which is good news. The bad news is that somewhere around December to mid-June, they started to stay alive, at least for a little period. But you can see from this graph, so the blue up here is the control, so it stayed alive for the most part. But what you see is that it was dead, and then when we would re-inoculate these for the cycling, you would see it start to kind of cycle around, and it would survive a little bit better. And so that basically meant that it can survive for longer periods from December to the mid-June time period, which is when we ended the study. So what did we find from a mutation rate? So obviously with the long-term ones, they died right away, so there were no mutations because they're not surviving for a long period. When it came to the cycling, we found that on average, O157 would accrue 1 mutation over this time period, so about 1, it equals out to a little bit over 1 per year. Typhimurium or Salmonella accrued about 1.5 mutations on average, depending on the environment. And Listeria averaged a little bit more in the control cycling, but it really didn't mutate a whole lot in the other environments. So the other thing the way we did this is that we did what's called shotgun metagenomics, which means that we took the samples after they were inoculated, we extracted all the DNA that was there, and we sequenced everything that was in there. So this also allowed us to look at the microbiome of the samples. And what we found along the lines was, I don't know how well this translates, but yeah, these are the controls. So this is the initial soil microbiome and what it looked like. And all you have to do is kind of look at the color bars. Here it is after 2 weeks. It's changed a little bit. This was after we put O157 in, so this big gray is actually O157. But you can see that there's an overall change in the profile, whereas you've got Salmonella and Listeria, and you don't see that change. So what this told us was that the inoculation of O157 into the soil actually changed the microbiome that's in that soil. We're still in the process of looking at how that happened and what happened and who was positively and who was negatively correlated, and whether we can use that for future work with food safety is yet to be determined. And that'll lead to what I'll end with. So ultimately, kind of what we concluded from this study was that there was a window for pathogen survival, and it really picks up from December to April. So early, as you're planting and things like that, there's less of a concern of the pathogen surviving. But as we get into those actual periods where plants are being harvested, and through the end, that's unfortunately when those pathogens can survive a longer period, at least in the soil and the irrigation water. There weren't any mutations from the long-term survival because they weren't surviving long-term. When we cycle them around, they'd accrue about 1 mutation per year on average. What does this translate into in the real world, which is the important part? What it shows is that cycling is important, and this correlates to the 2018 0157 outbreak. That one has been found in tons and tons of different sources, and it's slowly mutating, which correlates with what we're seeing, where when it's moving and cycling through these and getting into different environments, it's accruing those mutations. So that means that it has the potential of coming from a lot of different sources. Compare that to the 0157 strain that's been associated with the Salinas growing area for the last few years. This one is not accruing mutations, which means that it's probably sitting in some environment, not moving around, and therefore it's not mutating. Unfortunately, it's still getting onto crops and causing problems. So really that cycling is important in order for these pathogens to mutate. And so the other thing that we learned from it is that the pathogens, their presence in different environments can alter the microbiome. And real quick, I'm running out of time. That led to the current study that we're working on that's funded by the Arizona Department of Ag, which is to start exploring the microbiome and the interactions that the pathogen 0157 has in the microbiome, the soil chemistry, and things along those lines. So we've just began this. We've run one set and you can see we've also looking at the addition of soil amendments and irrigation water. Currently, what we've learned so far, like I said, we're only just getting into it, is that the pathogen, and this is grown under January conditions, so it's those ideal conditions, is that soil amendment actually helps keep the pathogen alive longer. It almost doubles the rate. And the irrigation water, surprisingly, didn't have as much of an impact as we thought. And Yuma, it did survive almost to 120 days. Compared, we're also doing this. There. All right, well, I'm just off. So it's also doing it in Maricopa, which it actually dies quicker in Maricopa conditions. And I skipped over my acknowledgement slide, but I just wanted to acknowledge particularly CPS, Arizona Department of Ag, for funding both of these, particularly since they're both in the room, but I would have acknowledged them anyway. Chana Rock, who's the rock star that she is, and everybody has mentioned her like 140 times. And then also the other one I want to mention is Victoria Oberg, who's my lab manager, who's done all this work.
Okay, next we have Alex Putman, and he is an assistant professor of cooperative extension at the University of California in Riverside. And Alex is someone that I collaborate with quite a bit on Fusarium wilt lettuce. He's a plant pathologist, and he's going to talk about pathology research in the California desert.
All right, thanks, Stephanie. Good afternoon, everyone. How's everyone doing? Good. Yeah, it's good to be here and see all the innovation, the growers in this area, and all the research here. And we saw an example of that in the lunch line today when the growers saw an obstacle that there was only one line open. They opened up the other side of the buffet line. So that was a good example of that today. Okay, so I'm just going to give a brief overview of my program. Work in two main areas that are relevant to the desert, one in downy mildews of a few different crops, and then soil-borne diseases. And so just getting right into it, going to blow through a few examples here. For onion downy mildew, they've worked out some epidemiological models that show the temperatures and relative humidity, those types of conditions that are favorable for disease. And so we thought to see if that works here in the desert. So we put small plot trials, we put a weather station in there because these conditions have to be measured in the canopy. And so we timed fungicide applications based on that. And so what we're seeing here is that we have maybe a standard calendar, we have a couple sprays, and these spray timing here was based on those models based on that weather station in the field. And then we have non-treated, and so we have this one application provided similar levels of control to this multiple applications. And so the way we set this up, we kind of package these models into a program, and the weather station uploads the data automatically to the cloud and is processed by the program, and then it sends out an email alert to us. And so right now we can at least, this onion's not too relevant over here in Yuma, but in California we can set it up for growers to receive these emails based on our weather stations. In fact, there is a grower here. There's a little bit of production that a processor grower is using this here in Yuma. So we can receive those emails every day based on the weather stations in the field. And so the vision for this is we want to take it so that you don't have to have a weather station in a field, and to use different sources of data such as weather to kind of to determine these conditions in the crop. And maybe you can have remote sensing or kind of these advanced weather models, so we can and build a system so that growers can have information about the conditions in their field, about the favorability for downy mildew. Another area of downy mildew was spinach. Ali Montazar, a water irrigation advisor in Imperial County, saw a grower who was growing spinach under high density, under drip. And so he initiated a project to look at a few aspects of that, and one of those aspects was downy mildew, which I collaborated on. And basically the bottom line is because you're not sprinkling, you're reducing the leaf wetness, and that's a huge driver for downy mildew. So see these two treatments on the right were produced under sprinkler through the whole, through the whole crop. And then these others were actually, some of them were germinated under drip, and all of them were maintained under drip after establishment. And not a whole lot of statistical differences, but there were areas of the field or these plots that had a lot of downy mildew. And so, again, this is driven by reduced leaf wetness, and this was organic, no fungicide applications, and also a highly susceptible cultivar that had no resistance genes. So again, this is something that probably be more valuable for organic, but driven by that reduction in leaf wetness. And that's an aspect of these models that I'm interested in, and that's the toughest obstacle to be able to deploy this widely, so to provide growers information in their field, because otherwise you need to have a sensor for this, and you probably need multiple because it's kind of a highly variable a thing to measure, and it's kind of difficult. And so that's something I'm interested in research-wise to make that more applicable. Okay, and so other aspects of downy mildew, participate in a project led by Steve Klosterman, who's a USDA scientist in Salinas, to trap spores and to kind of see the dynamics of these over the season. And not going to go too much into this graph, but we're looking at 3 different valleys, Salinas, Coachella, and Imperial, with both spinach and lettuce downy mildew. Of course, these are host-specific pathogens, so they're different species here. And so basically in the desert, we weren't seeing a huge numbers, of kind of close to not detectable, until we start getting a lot of crops. So maybe around this time, this is data from last season, we didn't really see the numbers increase until about this time last year. So when there's a lot of crop in the ground and it's starting to be harvested. And so this is another aspect because, of course, in addition to the environment, you need the pathogen to have disease. And so as another tool that I'm envisioning that we could provide the industry is to have some kind of tool that will give near or real-time information on the spores in the air. And so that could help growers make decisions on whether to apply fungicides or not. And there are some, what we're looking at it for onions, some lower-cost spore traps that's kind of really passive, and but now you have to send it off to a lab to do, but to assess that sample, but trying to look at other ways to make that faster and more accessible. Okay, and so that kind of puts it together, kind of what I just said, but we're also interested in looking at some other sources of where the pathogen is coming from, because downy mildews favor cool and wet conditions, of course, and so maybe the suggestion is they don't survive well in the desert of California, Arizona, and so we're wondering, well, where's the inoculum coming from? Where is that pathogen coming from to start those initial epidemics? And these pathogens can produce spores that can survive in the soil. They've started to show this in places like Canada. That's the source of that pathogen, and so we're interested in looking at that here, and maybe for things like, is it found on seed, and is that a source of inoculum? So because I'm trying to figure out if it can survive the summer, and then try to identify where those sources are and what we can do about them. Okay, and next is lettuce fusarium wilt is that main soil-borne project that I'm working on, and just to obviously it's a huge problem down here, but it's becoming a lot more of a problem up on the coast in Salinas, even though it's not a it's a much different environment and doesn't have that heat that traditionally thought of as the main driver here. And so starting to look at that as well. And one of those aspects is, as was mentioned earlier about in the industry panel, are there new strains of this pathogen out there? And from what I understand, with this, we haven't found anything down here, but up in Salinas, we have indications there may be a strain, and so here, a new strain. So we have here at a location that growers and seed companies and breeders have observed that this is their expected reaction for race 1 based on they know how the cultivars perform. And so just to back up a second, these were trials that seed companies, seed distributors, and breeders put out, and so this is something that they were noticing and they brought to us. And so we have on the left we have 2 susceptible cultivars, and they're susceptible at both of these locations, and we have a couple cultivars that are doing well, very highly tolerant. But down here they noticed that it has some trials at different reaction. Both those 2 susceptible cultivars are again susceptible, but the cultivar of those 2 on the top that were doing well are dead down here at this other location. And whereas there's some others that died in that first location, but that are doing better in this other location. So this is a pretty clear indication there may be something different there. And so we're characterizing those populations part of this project that Stephanie mentioned, and she was really the one who brought me into this. We kind of didn't expect to really find anything initially, and so we were already kind of geared up, and we're working on characterizing these populations. But again, she was instrumental in bringing us together, and so bringing this team together. But we meet regularly, and the team includes breeders, pathologists up in Salinas, as well as extension throughout the coast. And if there's anyone who has any connection with operations in California, just wanted to put out a request that we're looking for samples of this disease from Imperial Valley specifically. Stephanie's gotten a lot of samples here, but we don't have a lot in Imperial, so if anyone has any connections there and knows of any instances of this disease, we would appreciate it telling me. So yeah, so thanks again. This has been a good meeting. So does anyone have any questions? Yes.
So on the picture with the possible different rates, are those wet on 2 different dates, or were they
Those are 2 different fields.
2 different wet dates, but are they close in time?
Oh, in terms of maturity? I don't remember the exact dates, but they were, they're pretty close in terms of maturity, those 2 different photos, yeah. Any more questions? All right, thank you.
Thank you, so I'd like to thank the organizers for the opportunity to speak here. I will have a little bit of an explanation about what a fundus is. It doesn't mean fund us, although I think that's quite curious, and I like the word for that reason. But, I'll speak today a little bit about an endeavor on which we're embarking now, which is a lot like what you described, Duke. We'd like to do it in humans, but we can't make 20,000 people stand in one place for 2 months and, pass the sensors over. So we'd like to involve these mobile sensors, and that's a lot about the project that we're speaking, that I'll speak about. So we talked a lot about industry partnerships. I'll start by first describing a study as an industry university collaborative. We have our director and 2 colleagues here, and I'll introduce you guys later, but it's something that we think can be well informed by what we learn from agrotech. So I'm just stating the obvious to say that the quality of food is something that, and the nutrients and the bioactive constituents of food and the value of food sustains life. And we all knew this, but I guess the point is, if we are moving towards a place where we can assess the abundance and the speciation of certain nutrients that are important for key physiological processes in the human body, but also in plants, in animals, in microbes. We've talked about all these forms of life. If we can sense that and optimize that sort of thing, we can make the health of the planet a lot better. So, okay, so let me just figure out how to advance this. Okay, so fundus. So the fundus is basically the back of the eye, and I already made that joke, but so I'll just move on. I'd like to speak a little bit about this government, industry, academia collaboration. We are a collaborative research center. We focus on sensing and signaling of living organisms, decision systems. We use an approach called bioconvergence, and Parker was referring to this as convergent thinking, and the purpose is to move towards health equity, to get to people and spaces in the world that have been marginalized, that actually need some help in either survival or rescue of certain aspects that would keep these systems going, these essential systems for life. And again, I talked a lot about how this relates to planetary health, population health, and eventually personalized. So, to get back to what Duke was speaking about, we're very interested in assessing the variation and complexity in these ecosystems and what we can actually modify. And we're going to do that through sensor technology. So we have a group that includes the University of Arizona, Baylor College of Medicine, the whole Houston medical area, Caltech, University of Missouri, North Dakota, and USC. So we're focusing mainly on humans, but we'd like to talk to people here about what you know and how we might be able to partner on the front end, and I'll get to that. So, okay, so about bioconvergence, this is sort of a history of my family. My grandfather was a farmer. These are his brothers. He did wear a tie when he was farming. I noticed that was quite interesting. So I took my tie off today, but the next generation, my dad and my uncle were engineers, and I think I'm a nutritional scientist, and my son is a chemist. So we need to sort of put this together in real time and see what's happening at the moment. So right now, the team that we have here today involves an engineer, which is Dr. Roveda. And if you could just raise your hand. So she's our PI, and she knows way more about this study than I do. We have Dr. Wang, and she's a clinician, but she also grew up on a farm, right? So she's the only one that has experience farming. And we have Alex here, who is an expert in nutrition and mineralogy. So you can see that we have this multidisciplinary team. There are many other people in the group. These are the people currently at the University of Arizona. I might just go through this quickly, but I thought about the alignment in the mission of what we're doing here today and what we're doing with the NSF, and that could be related to what was referred to as an interagency agreement. So Andy, you talked about that today, and what we're hoping is that, again, we can put this front end on what we consider an important exposure in terms of nutrient density, the quality, the distribution of nutrients in our food, and how we can work with agrotech to make that work. So again, we're also looking for partners. Anyone here who's interested in this? Okay, so we have a use case here, and this is based upon some research that I've been doing for about a decade and a half. I'm very interested in carotenoids that are preferentially deposited in our retina. And so these carotenoids have bioactive properties that actually protect us, but the idea is if we can actually eat the right kinds of foods or make the right kinds of foods and distribute those, then people will have higher concentrations in their retina. But the basic axis is that we look at the intake. There are different ways that we measure response. These are images from my laboratory, so we do live imaging of the retina. This is actually my retina. And then we also have physiologic measures. Okay, so to go back to this large collaborative, what we're doing is we're developing assistive devices, remote monitors, digital treatment, and diagnostic technologies. We have tools to increase the efficiency of workflows and decision support. So that was our device and sensor. We also have this aspect of partnership. We've talked a lot about this. And then we have this interface between the provider and consumer. My son said these look like lawn darts, but I told him it was Arizona, and you can't really use lawn darts in Arizona because they just bounce off the ground. So, but basically, you know, we have these three domains. We're very interested in designing, testing, and what we're calling streaming information. And the streaming part is related to teleconnection. So what we develop are wearable sensors or sensors in place. And then this is basically geared towards human health, but again, we're interested in what happens after an exposure. And we're doing trials and demonstration projects in the same way that you do. The value to our industry partners is that there is a return on investment because you can leverage the strength of the university system, access to our technology, and then we have training programs. And again, we're using the Internet of Things, AI, and big data for decision sciences. So what does this mean in the context of what we're doing here? We have posters. Dr. Schaus has one on specialty crops that are grown, traditional crops that are grown in arid environments. We have a multispectral imaging device that is can be applied in food safety monitoring. And then there was some information on bioactives and actually extracting nutrients from crops for the purpose of developing nutraceuticals. We haven't talked about that today, but Shane, you talked about nutritional products, and this is a possibility that we see as maybe a spin-off market. Okay, and then I'd like to also talk about our personalized and human nutrition approach. So this was the axis that I described, but if we think about what you're doing here, the sustainability and the quality of the nutrients that are resident in these foods that are enriched in the retina is something that I think we can probably work together on. I think it's important to think about that. So there are 600 naturally occurring carotenoids, and that's what I'll talk about next. There are 50 in the diet, there are 20 in the circulation, and there are 2 in the retina. Did anyone already know that? I'm just wondering. Yes, and did? Good. I'm not surprised. Okay, so they're known as lutein and zeaxanthin. Now, the thing is, we have about a 10,000-fold increase in the retina relative to the next most concentrated tissue. So what might that be about? I can skip through these, but basically we have to think about what the properties of these nutrients might be. They basically interact with membranes. They act in the attenuation of energy. They can modulate, they can act in oxidative stress pathways and redox balance to accept electrons and reduce this oxidative stress. And finally, they can act to bind transcription factors. Okay, so I talked a little bit about the macula. I'm low on time, so I'm going to try to go quickly through this. But we know that if we think about how we concentrate these nutrients, it's an active process. And we know that it's a direct uptake by certain receptors. And if we look at the concentration that we can get through the diet, we're not nearly getting enough. So the daily intake is about 2 milligrams, but we can safely take 75 in, and that's for the lutein. For the zeaxanthin, it's about 0.2, and we can take up to 50. So the idea is if we can use these in vivo imaging technologies, and we have these, this is an autofluorescence method, this is an image of the retina, and we can see the spectrum, which represents the optical density or the concentration. Okay, sorry. And this is a Raman resonance image, and we can see that those are clearly indicating that we have high levels. I'm going to skip through this part just to the very end, and this is the part that relates to what Duke's doing. So what we're doing, again, is we're embarking on an endeavor right now to develop sensors, mobile sensors, so no one has to stand in a field, okay, to measure what we're calling an exposome, of which food is part. And then we're going to look at all these factors that affect the distribution, the accretion, the transport of nutrients in the body. But because we have information on the genome, we can actually look at individual variation and complexity. I think that's I'm probably close to being out of time. Yep, okay. And we can do the same in plants, but I just wanted to let people know that we're available to speak. And again, the people in my group, if you just raise your hands, we can maybe meet after in the time afterwards. Thank you. Okay.
Thanks. That was a great talk. Our next speaker, our last speaker, is Pedro Andrade Sanchez. He's an associate specialist in precision agriculture at the University of Arizona, and his talk is In-Field Soil Variability in the Yuma Area, Tools to Characterize It and Implementation of Variable Management Technologies.
Good afternoon, everybody. My name is Pedro Andrade, and I'm really happy to be here. Thank you, Stephanie. Thank you, Paul, for the invitation. So today what I'm doing is presenting to you about my work in this area that is heavy on machine systems. I'm going to use this picture to show you really the context of my talk. First of all, I want to introduce John Hune. That's the guy driving that tractor. He and I work on these materials. But an important point for those growers in the room is to mention that this is what we do for research, and this is how close we are to put these tools in the growers' use. So for casual observer, that looks nothing different than any tractor working in the field. Okay? And that's how we like it. That's how we want it to happen. So I'm going to get in my talk first with, I want to make sure that I convey the message of what is soil variability and why it's important and why we need to pay attention to it. So I have three different scales to show you relevant points. So I don't know how well you can see this map. This is one of those USDA NRCS maps of soil maps. And in this case, I took an area here all the way to close to Somerton and here. So in this area of about 21,000 acres, there are 13 major soil units. And out of those 13, I'll say 10 of those really have a significant area. And if we click on any one of these, you'll see all the details that define those units. But let me tell you, they're very different from each other. And there are fields, look in this area, look at this, that those are mixed, showing you that it is very difficult to characterize what's the dominant soil characteristics in many cases. So that's one scale to look at variability. Now, there's another scale that I want to present, and this I'm using 1996 Google Earth image. This is the UMass Center. And in this case, what I notice is that the image comes from the USGS, the United States Geological Service. And for some reason, I consulted this with Andy French. So these images that are very old, they show features of the soil that are related to soil variability. So again, I'm at a much finer scale than the first map. And at this scale, I see all kinds of different features that are soil related. These fans of sandy soils, very much connected with the soil formation processes here in the desert. So these will have very significant effects on how the crop responds to those changes in soil condition. A third level, this is the finest level that I'm presenting to you today of soil variability, is this particular field in the Somerton area where we harvested back in June. And in this case, you can see that the distribution of values can be as low as 5,000 pounds per acre to all the way to 8,300 pounds right here. Uniform management. There is no other reason to allocate, to explain the variability in yield production other than soil differences, okay? So with that, what I'm saying is that it is quite significant. Regardless of the scale that you look at, it is a significant amount of soil variability. That tells us that uniform management is one thing, is the easiest you can do. You go for the average, kind of, and not quite. I'm going to show you that actually in Yuma, we are not doing the average. We're going in a different direction. Now, this is the same area, Google Earth, but now I'm looking at November 2017. And in this case, it's November. It's expected to be a lot of crops, a lot of vegetables during this time frame. And I don't know from your angle, this doesn't look the way it looks in my computer, but I don't see much variability this time around. It's almost negligible. All I see is green, different tones of green, different crops, but very little. It's not as, oops. Certainly not as, there's a big contrast in terms of variability in these images. Okay, so here's the point that I want to bring. This is a digital image. It's quite different in terms of the type of image that is here. The point that I'm bringing here is that there may be many reasons for this lack of variability in this image, in these fields, but one that resonates with me is the possibility that we are over-fertilizing our fields. So in my opinion, what we're doing in Yuma with the cost of fertilizer materials is to go on a safe side and apply more than what's needed, okay? I don't have the data to suggest that, but I have enough information to make a point. So let's go for that. This is a piece of research by Charlie Sanchez and in Yuma. This is water and nitrogen relationships in terms of productivity of lettuce. So starting here, you see that in these two axes is always water used and nitrogen applied in all three of them. So I made a conversion, and this is where we look at an optimum point in this surface. And Yuma growers are very good at reaching this point. They've been doing it for many years. Yuma growers are really an example of growers that work on optimizations. Now, looking at nitrogen use, in this case, you see the nitrogen that was not recovered out of about 300, if you follow this line here, it lands on about 300 pounds per acre. Kilograms per hectare is very, very close to pounds per acre, just so you know. So 187 are not recovered. And my understanding is that 134 that are leached are part of this. Don't know for sure, but Charlie is the one that can answer that. But the point is, if this is what's not recovered, and this is what was applied, we're losing about 62% of that nitrogen that was used. Gone, not used by the crop. So that's what I'm telling you. There are some indications that indeed we are over-fertilizing. And we know that we need to have a crop that is green, that has good market quality. That's beyond the question, is that we have to keep that quality. But really the point that I'm bringing at this moment is that there is technology to improve this. That's the basis of my talk. So with Charlie Sanchez, we've been doing a lot of work in the Yuma area on variable rate nitrogen management for the last 3 years. And I'm going to show you a few things that we've been doing. Sometimes it's a blessing not to have money because that forces you to innovate and try to do something with what you have. So we came to this project with funding for do our field work, but no funding for any capital expenses. So what we did is to use this frame that came with these probes, instrumented with LED-based soil spectrometry sensors and thermistor and a capacitance-based soil moisture. And John, what he did is that he integrated all the hardware, and then he found a way to make the system to emulate an operation that is actually not happening, but that enabled the data acquisition system, and that's how we collect data here. So we found a way around it, and this is our frame. This is what we're doing to collect data georeferenced. In this case, you can see that this is during or before, during soil preparation, before there is a crop. So very quickly, these are 5 different soil parameters. How much time? Oh, should I stop then? I'm sorry. 5 parameters that we collect simultaneously, and this is the rate of work that we have that we can reach. This is another look at the different frames that we use for the application. Everything that I'm showing here is application only, and these are the numbers to show you. This is really information intensive. That's the only difference with a conventional frame, that ours have a lot of information inputs. I'm going to skip that one, and I'm going to say on this one, this is what we've been doing in terms of soil sampling, the soil sampling approach to do variable rate. And we are relying on these functions that are very well known, what's in the soil prior to site dressing, and what is the recommended rate translated to UAN 32. So we know that. We know for certain crops that is slope, and the offset tells us something about these functions. I'll describe in a moment. But a key point to transmit today is that with conventional rates that we use between 30 to 40 gallons per acre of UAN 32, that satisfies this bar here in this histogram, which is about 28% of the samples that we collected. Actually, growers are under-fertilizing 9%, and the rest, 62%, they are over-fertilizing. That's the inverse relationship that you can see in this graph. So I apologize for not measuring my time. But on this one, all I want to say is that actually Yuma is a very special case for machinery use. And these machines are here, and whether they are used in whatever mode of operation, they are capable of doing variable rate. The software is loaded, whether you use it or not. I had 5 summary points to make. I guess in the interest of time, I only want to talk about number 5, and that is, that's what we can do. I had the goal of presenting to you what is it that we're capable of doing, how we can integrate systems, how we can expand our spatial analysis. But in the end, that's all we can do. We need to extend that support in areas where our engineering cannot go any further. Okay, so that's where we stand.
I just want to mention that, and this was mentioned before, but this symposium was supported by an SCRI planning grant. The title was Developing a Transdisciplinary Approach for Improving Leafy Greens Production in Arid and Semi-Arid U.S. Growing Region. We got the money right before the pandemic, and so much changed about it, but we're glad that we actually got to have this in-person event. The objectives were to identify the needs of the industry, and we did this by a survey and getting feedback from experts, and there's a survey document on the registration desk that you can grab if you're interested. It's it's research priorities, so it's identifying the highest priorities of the industry. And building collaborations and teams was one of the big goals of this project, and from all the conversations that I've been part of, I think that we've gotten really far in the past 2 days that we've been together. Some of us have been together, but even before that, there's been so much team building over the time of the pandemic because we were having Zoom meetings. There's been lots of project planning. I'm really happy with how far we've gotten the past few years. And now we're on the addressing needs part. So I hope that we get some new projects out of this. I just want to thank you all for coming, and thank you for the people who helped sponsor the event. And I hope to see you all again in Yuma soon.